The effects of rainfall on E. coli measured in Waller Creek. This experiment has 3 possible conclusions in determining whether or not rainfall has an effect on E. coli in Waller Creek. The levels of E. coli, in response to the number of days since the last rainfall, could decrease as the number of days increases, increase as the number of days increases, or see no change in response to rainfall. So what is E. coli? E. coli is a rod-shaped bacteria commonly found in the gastrointestinal tract and feces of warm-blooded animals. It is a member of the fecal coliform group of bacteria and is distinguished by its inability to break down urease. It is usually harmless and actually aids in the digestive process. However, E. coli can cause illnesses such as meningitis, septicemia, urinary tract, and intestinal infections. In severe cases, certain strains of E. coli can cause severe disease and can also cause death in small children and the elderly. How does E. coli impact streams? Its presence suggests a possible fecal contamination. This can be the result of dog parks, livestock runoff, or even a sewage leakage. Despite this, it is actually a very natural component of rivers, lakes, and streams. However, high levels of measured E. coli in a given water source may pose health risks to humans. So, how does rainfall affect the presence of E. coli in streams, and why is this important? In this experiment, 2 data points will be taken from 5 different sites along Waller Creek, ranging in time since the last rainfall. These 5 locations are Palm Park, abbreviated as PP, 10th Street, abbreviated as TS, Medical School South, abbreviated as MS, the UT Clark Field South, abbreviated as CF-S, and the Creekside Dormitory, abbreviated as CS. At each of these locations, the 2 data points taken correspond with the number of days since the last rainfall. The first set of data will be recorded 24 hours after the last rainfall, while the second set of data will be recorded 48 hours since the last rainfall. This data was collected by the 2 lab times which had divvied themselves up into teams of 2. One partner recorded the data from the YSI and took pictures of the surrounding area. The other partner prepared the 500 milliliter sample bottle by rinsing it 3 times with creek water, mostly filling the bottle with each go. On the 3rd rinse, the contents of the bottle was not dumped out, and instead this became the water sample for each team. Materials and methods. This experiment began with sample collection. This was accomplished by filling an amber-colored 500 milliliter bottle with water from each of the 5 locations at their respective times. From there, the samples were brought back to the lab, where dilutions of varying PBS and creek water were created. These dilutions were then placed onto 3 petrofilms, which were then placed in an incubator that kept the samples at a constant 35 degrees Celsius. After 24 hours of incubation, the number of bacterial colonies that formed on the petrofilms was recorded and converted into CFU per 100 milliliters. As for the safety precautions that were followed during this procedure, at each of the sampling sites, we were monitored by a lab curator, and we wore proper gear like gloves or long pants. This was intended to minimize contact with the water, as it is harmful to humans. These safety procedures also continue into lab as to reduce any risk of contact with the bacteria. After the proper calculations were made, the graph shown displays the recorded E. coli and CFUs per 100 milliliters at each of the 5 locations. The y-axis displays the measured E. coli, while the x-axis depicts the abbreviated locations. At each of these points, there are 2 data columns. The blue column on the left is representative of the data collected 1 day after the last rainfall, while the orange column on the right depicts the data recorded at the same site 2 days since the last rainfall. As showcased in the graph, there is a clear difference between the data collected at these 2 times and is consistent across each location. So, what do these results mean and what conclusions can be made? After performing a one-tailed paired t-test A p-value of 0.01908 indicated a strong statistical correlation between E. coli in a stream and the days since rainfall. This provides mathematical backing to the visual correlation depicted in the graph on the previous slide. As for conclusions, in the day immediately after a period of heavy rain, there will be higher levels of recorded E. coli. As the number of days increase since the last rainfall, the measured E. coli in the stream will decrease. These results were expected due to reasons addressed in the next slide. What causes this trend? As rainfall occurs, it creates runoff. This runoff transports any surface pollutants into local streams. The main culprit in surface pollutants that contributes to the amount of E. coli in a stream primarily consists of fecal matter. As mentioned earlier, these sources are most often humans, pets, livestock, and wildlife. But rain also has another effect on streams and water sources. This is erosion. As heavy weather systems occur, they cause a great upheaval and movement of sediments as well as other particulates. This in turn contributes to another situation, turbidity. Turbidity is the measure of relative clarity of a liquid. It is an optical characteristic of water and is a measurement of the amount of light that is scattered by a material in water when a light is shined through the water sample. The higher the intensity of scattered light, the higher the turbidity. This is beneficial for E. coli growth, as it decreases the amount of sunlight able to permeate through the creek. Sunlight is harmful to the bacteria and destroys colonies. Until the sediments have settled and turbidity lowers, E. coli will experience an elongated period of growth as a result of rainfall. Why is this important? The presence of E. coli in streams, as mentioned earlier, elicits health risks ranging from minor symptoms of diarrhea to death. Its presence in streams and other bodies of water limits the utility of these sources. For example, for water to be used in direct human consumption or drinking water, there must be no E. coli colonies present. Another property of water that is beneficial to humans is recreational activity. This is any activity in which skin contact is made with the water. This can be swimming, sailing, water skiing, etc. E. coli levels that become too high will start to cause issues even with skin contact. Thus, in these cases, no human contact can be made with that particular water source. E. coli can also cause concern for domestic animals that might interact with a water source. This may pose health risks to pet owners or dog parks. It may also threaten livestock. This would not only elicit a field response from the public, but could also negatively impact the agricultural economy. On a more optimistic note, further research can lead to discoveries regarding sources for E. coli contamination. With this, we could isolate specific contributors of E. coli contamination and either remove these sources or enact regulations to limit their effects. Ultimately, regulations can improve water quality and stream health. Waller Creek, with enough dedication and revitalization, could see a reduction in E. coli colonies and an improvement of its urban environment. It may one day be available to recreational public use.